For an organism defined by the physics of flight, every milligram of weight and every point of aerodynamic drag is a survival factor. This necessitates a body plan that internalizes reproductive hardware, moving it away from the external environment. The avian male reproductive tract is entirely internal. No scrotum, no external testes. In this drawing of a rooster's dorsal body wall, we see the paired reproductive tracts packed tightly against the internal cavity. Notice the paired testes positioned precisely adjacent to the cranial lobe of the kidneys. The deferent ducts run alongside the ureters to the cloacal opening. In many species, the left testes is larger than the right. This is a functional, natural asymmetry, rather than a developmental flaw. This internal architecture trades the specialized cooling of a scrotum for aerodynamic efficiency, forcing the avian system to perform spermatogenesis at core body temperatures. Avian testes exhibit extreme volumetric plasticity, expanding or contracting based almost entirely on the duration of light exposure. During the short days of winter, the testes remain small, pale, and completely regressed. But as spring approaches and daylight increases, the organ responds. They undergo a 200 to 500-fold volume increase, reaching peak reproductive capacity the moment a critical spring daylight threshold is crossed. To understand how this growth occurs, we look at the microscopic architecture of the testis, specifically the interstitial space and the seminiferous tubules. In the winter state, the tissue contains abundant interstitial space and narrow tubules. As the organ grows, the relative volume of interstitial tissue shrinks, and the seminiferous tubules expand to dominate the organ. Light egg cells produce testosterone. Sertoli cells nurse developing sperm. The two-cell model of testicular function applies in birds as in mammals. The bird rebuilds its reproductive capacity from the ground up every year, a strategy that preserves vital energy during the non-breeding season. The duration of light in a given day dictates the transition between reproductive states. Long days initiate the activation sequence, while short days bring it to a halt. Environmental light penetrates the skull, directly stimulating deep brain photoreceptors. These trigger the hypothalamus to secrete CGNRHI, avian reproduction's prime regulator. The pituitary responds by releasing two gonadotropins, LH and FSH. LH impacts Leydig cells, surging testosterone, while FSH hits Sertoli cells, driving rapid cellular division. When days shorten, the hypothalamus begins secreting a dodecapeptide, known as GNIH, or gonadotropin inhibitory hormone. GNIH actively suppresses GnRH, blocking the release of LH and FSH. This leads to gonadal apoptosis and seasonal regression. This push-pull neuroendocrine control ensures that the energy cost of reproduction is only spent when environmental conditions are optimal for chick survival. Inside the active testis, sperm is manufactured within the seminiferous tubule's lumen. The meiotic sequence begins at the basement membrane with the mitosis of ADAD and AP1 spermatogonia. As these stem cells divide, they push inward toward the pale blue center. As the germ cells separate from the basement membrane, they transform into primary spermatocytes, then divide into secondary spermatocytes, and finally into haploid spermatides. This initiates spermiogenesis, the final phase where spermatids transform into mature spermatozoa without further cell division. This involves a drastic physical reduction. A mature rooster sperm embodies only 3% of its initial spermatid volume. To achieve this, the cell sheds its cytoplasm and undergoes severe nuclear condensation, replacing standard nucleohistones with nucleoprotamine. Once packaged, the mature spermatozoa detach into the tubule fluid, ready for transport. Spermiogenesis in birds strips away non-essential biological components to create a hyper-light genetic payload. This scanning electron micrograph shows that avian sperm have a distinct morphology, a long helical head compared to the paddle-shaped head of most mammals. The helical head is a defining feature of avian sperm, reflecting a different design from most mammalian sperm. In species with intense sperm competition, like passerine songbirds, this helical shape becomes even more exaggerated to outcompete rival males. Newly formed sperm exit the testis and enter the epididymis through the lacunae of the rete testis. As this data table shows, the avian epididymis is highly condensed and is dominated primarily by the efferent ducts rather than the epididymal duct. The avian epididymis is structurally simpler than in males, but remains functionally essential, sperm leaving the testis are not yet capable of fertilization. Despite lacking the massive storage capacity found in the mammalian epididymis, this abbreviated duct system is required to biochemically mature the sperm and grant them the potential for motility. From the epididymis, sperm enter the convoluted ductus deferens, where seminal plasma is introduced. Birds lack accessory reproductive glands like the prostate. Instead, the ductus deferens acts as the primary storage vessel and the producer of seminal fluid. In this cloaca schematic, the deferent duct widens into a receptacle. Most birds lack a phallus, relying instead on a brief cloacal kiss. The avian erection mechanism is lymphatic, not blood vascular. The paracloacal vascular bodies create lymph to engorge the cloacal folds. Because there is no internal urethra, semen is transported across the tissue via an external groove called the sulcus spermaticus. This entire system, internal, photoperiodically controlled, and utilizing a lymphatic delivery mechanism, represents an optimal physiological compromise. It ensures reproductive efficiency without sacrificing the aerodynamics required for flight.